Hello and welcome to this video on protein degradation and ammonia metabolism. As medical students, understanding these concepts is crucial for your biochemistry studies, especially when preparing for the USMLE. My name is Dr. Morabais, and I am a professor of biochemistry. I have been teaching biochemistry at medical schools across different continents for about 30 years. In this video, we'll cover the key areas of protein degradation, deamination reactions, transamination reactions, ammonia production, and ammonia transport. In a nutshell, what we will cover in this video is displayed on the screen. Protein degradation is the process by which cells break down proteins into smaller peptides and amino acids. There are two main pathways: the ubiquitin proteasome pathway and the lysosomal enzyme degradation pathway. Proteases are essential enzymes that catalyze the breakdown of proteins into smaller peptides and amino acids, aiding in various biological processes. Protein half-lives vary, and several factors determine them. For example, the amino acid occupying the aminoterminal position and the occurrence of PEST sequences are some of the factors that determine the rate of protein degradation. The ubiquitin proteasome pathway is responsible for degrading damaged, short-lived, or misfolded proteins. It is ATP-dependent. Long-lived protein, membrane surface proteins and extracellular proteins are degraded in the lysosomes. Lysosomal degradation is very essential for recycling cellular components and regulating cellular metabolism. It doesn't require ATP hydrolysis. Now, let's look at deamination. Deamination is the removal of the alpha amino group of the amino acid, leading to the formation of the corresponding alpha-keto acid. Oxidative deamination utilizes amino acid oxidases which require oxygen and utilize flavin nucleotides as coenzymes. Non-oxidative deamination do not require oxygen. Role of deaminase enzymes. Deaminase enzymes play a crucial role in catalyzing the deamination of amino acids into keto acids, facilitating metabolic processes. Nitrogen excretion. Deamination is vital for nitrogen excretion, helping to maintain nitrogen balance in the body and prevent toxicity. Energy production. The conversion of amino acids to keto acids through deamination contributes significantly to energy production in metabolic pathways. Oxidative deamination. This involves the reaction of an alpha amino acid with oxygen to form an alpha-keto acid and ammonia. Amino acid oxidases catalyze the reactions, and they require flavin nucleotides as coenzymes. Non-oxidative deamination does not require oxygen and is catalyzed by specific enzymes, e.g., serine dehydratase and threonine dehydratase, as displayed on the screen. Note: both serine dehydratase and threonine dehydratase require pyridoxal phosphate as coenzyme. Oxidative deamination reaction catalyzed by glutamate dehydrogenase is very crucial in the body. But, note, it is a reversible reaction. During the conditions that favor transamination and urea cycle, equilibrium favors the deamination of glutamate to form alpha-ketoglutarate and ammonia. However, during the conditions of elevated ammonia levels, equilibrium favors the formation of glutamate from ammonia and alpha-ketoglutarate. In addition to amino acid deamination, several other nitrogenous compounds also undergo deamination through various enzymic reactions. For instance, monoamine oxidase deaminates the monoamines, such as dopamine, norepinephrine, epinephrine, tyramine and serotonin. Purines and pyrimidines are deaminated by other enzymes. The deamination process is essential for maintaining nitrogen balance in the body, crucial for health and metabolism. Now, let's talk about transamination. Transamination reactions involve the transfer of an amino group from an amino acid to a keto acid. A new amino acid and a new keto acid are formed in the process. Some non-essential amino acids, such as alanine, aspartate and glutamate, can be generated through transamination reactions. Aminotransferases, transaminases, catalyze transamination reactions. They all use pyridoxal phosphate, PLP, as coenzyme. Aminotransferases contribute to metabolic flexibility by maintaining amino acid pools necessary for metabolic pathways. Notable examples of aminotransferases are ALT and AST. ALT, alanine transaminase, is primarily found in the liver but is also found in the skeletal muscle. AST, aspartate transaminase, is found in the liver and other organs such as the heart, kidneys, skeletal muscle and brain. Elevated levels of AST and ALT indicate organ damage. ALT is a specific indicator of liver damage. Here is a typical example of transamination reaction. Let's talk about ammonia transport. Ammonia production. Ammonia is produced as a byproduct during the catabolism of amino acids and other nitrogenous compounds, such as monoamines, purines and pyrimidines. Toxicity of ammonia. High concentrations of ammonia are highly toxic and can lead to severe health issues if not properly managed by the body. The brain is particularly vulnerable to ammonia toxicity, and the body has evolved mechanisms to transport ammonia out of the brain. The glutamine synthetase reaction in the brain converts ammonia to glutamine, which is then transported out of the brain. This reaction is crucial for maintaining brain health. Management of ammonia levels. The body employs various mechanisms to efficiently manage and eliminate ammonia to prevent toxicity and maintain health. Ammonia is transported from the brain and some other tissues to the liver or kidney via glutamine. However, ammonia is transported indirectly from the skeletal muscle to the liver via the glucose-alanine cycle. The glucose-alanine cycle is very important during fasting or starvation. When ammonia level rises, it combines with alpha-ketoglutarate to form glutamate through the reverse action of glutamate dehydrogenase. Alpha-ketoglutarate becomes unavailable for Krebs cycle, leading to low production of ATP. Troubles ensue, especially in the brain. Now, let's talk about the glutamine synthetase and glutaminase reactions. As noted earlier, glutamine is formed from ammonia and glutamate in the brain. The reaction is catalyzed by glutamine synthetase and it is ATP dependent. This reaction prevents ammonia toxicity in the brain. The glutamine comes to the liver or kidney and is hydrolyzed to glutamate and ammonia by the glutaminase enzyme. Note: in the liver ammonia is converted to urea. Watch my video on the urea cycle to get details of the cycle and its associated metabolic disorders. In the kidney ammonia, as ammonium ions, is excreted in the urine. Ammonium ions play a role in the maintenance of acid-base balance. Ammonia production increases in metabolic acidosis and decreases in metabolic alkalosis. For medical students studying biochemistry, it's essential to focus on the following high-yield areas: protein degradation pathways, including the ubiquitin proteasome pathway and the lysosomal enzyme degradation pathway; transamination reactions and the diagnostic values of transaminases, such as ALT and AST. Ammonia production and transport mechanisms, including the glucose-alanine cycle and the glutamine synthetase reaction in the brain. The roles of glutamine synthetase and glutaminase reactions in ammonia metabolism. The impairments caused by ammonia toxicity, including hepatic encephalopathy. Some key terms to remember and study for the USMLE: protein degradation, deamination reactions, transamination reactions, ammonia production, ammonia transport, ubiquitin proteasome pathway, lysosomal enzyme degradation pathway. ALT, AST, glucose-alanine cycle, glutamine synthetase reaction, glutaminase reaction, hepatic encephalopathy. Question for you. Why is the renal glutaminase reaction very active during conditions of metabolic acidosis and inactive in metabolic alkalosis? Drop your answer in the comment section. Thank you for watching this video on protein degradation and ammonia metabolism. Remember to focus on the high-yield areas outlined in this video, and you'll be well prepared for your biochemistry studies in the USMLE. Good luck with your studies. If you have found this video useful, subscribe to our channel to have access to our other exciting medical biochemistry videos. Until next time, bye for now.